Welcome back to the class. We are discussing about the plant tissue culture concept. As a part of it, in today's class, we are going to discuss about the lab facilities which are required to carry out the plant tissue culture experiments. As I have already stated you, plant tissue culture is a highly sophisticated process. It requires a highly skilled person as well as a well-equipped laboratory. So both the combination will bring out the success of plant tissue culture. So in the image we can able to see list of materials which we require to carry out the plant tissue culture experiment in a successful manner. Let us see what and all we require to carry out the plant tissue culture processes. First we require, so as it is mentioned in the book, we require a lot of items. Let us see few things which we need to carry out the experiments. First is here given as the transfer room. So this is a very important area where the explants are transferred to the nutrient medium. You can able to see the picture, right? So there were a lot of scientists, three different scientists were sitting in the front of laminar airflow chamber. So this is the laminar airflow chamber we can able to see. So this is the laminar airflow chamber. So you can able to see the filter sheet, right? So this is what we call it as HEPA filter. We can see in the next image. So through this, the fine hair will always flows, right? And here the scientist will sit and transfer the explant onto the nutrient medium. And this area is what we call it as a transfer room of plant tissue culture. And this is a highly sterile environment, and mostly we can't able to access in this room, right, without proper permission, right? This is a very important area in plant tissue culture lab. This is what we call it as transfer room, a highly well equipped and sterile room. And this is the laminar airflow chamber. We can able to see this. So here we can able to see in the laminar airflow chamber. So this is a glass door, right? So all the three sides are closed by glass wall, and you can able to see a filter here inside the room, inside the laminar chamber. We can able to see a filter, and that is what we call it as HEPA filter, high efficiency particulate air filter. And this filter is very important because this filter, HEPA filter, will filter the air, thereby filtering the dust particles, which are fine dust particles which are present in the air, is filtered by this HEPA filter, and the air which blows out is devoid of any sort of dust materials. So that is why we are using this HEPA filter. You can able to see here, and this is the HEPA filter, and this is the internal organization of HEPA filter. We can able to see multiple rows of filter papers are there, right? So while the air passes through this filter, all the dust particles, because these filter papers have very micro pores, and while the air passes through these microspores, what are all the dust particles it has, it will get filtered here, right? So that is how it works. And you can able to see here in the laminar airflow chamber, inside the chamber we have a UV light, right? So this UV light is used as a precautionary step. It is used as a to make the environment a sterile one, right? So UV rays, as we know, UV rays has the ability to kill the microorganisms, right? So that is why we use the UV radiations in the laminar airflow chamber. So you can able to see in this picture. So a person is using this laminar airflow chamber, right? So this is how the people will sit and make use of this laminar airflow chamber. And this is another room with regard to plant tissue culture, what we call it as sterile culture room. So this is the culture room. Once the explant is transferred to the culture medium, and it is transferred to this room, what is called as culture room. And in this culture room, we can able to see the entire setup is highly controlled environment. We can able to see the proper light facilities, air facilities, right? And the area, see, look at the area. So the entire area, the entire room is highly clean, devoid of any sort of dust or any unwanted things, right? So this is what we call it as culture room. And here, and this is the place where the culture are maintained, right? So this is the very important machine or the device which is used in the plant tissue culture, what is called as autoclave. So autoclave, this is nothing but a large size pressure cooker, right? So this autoclave works under the principle of a pressure cooker. So this autoclave is used to sterilize the materials used in plant tissue culture, more specifically the glass vessels, and then forceps, scalpels. All these are the materials that can be sterilized using this autoclaving. So we can able to study about all these sterilization process in the next video. So, and this is the lab oven. This is a large size lab oven which is used to, this is also used for sterilization process, and it is specifically used for dry sterilization. Once the vessels, the glass vessels, utensils are washed and they are kept inside the oven to kill the microorganisms, and using this oven we can able to sterilize in a dry environment. So the oven is used for dry sterilizing the materials. And this is what we know, this is the laboratory thermometer used to measure the temperature of the medium used. And here is the pH machine, and the pH machine is used to detect the pH value of the nutrient medium. And this is a well equipped pH machine, the latest form of pH machine. And the pH of the medium is always used to be acidic, that is 5.6 to 6. It will be between the range of 5.5 to 6. That is the most optimal level of pH for plant tissue culture. And you can able to see this electronic balance since while preparing the medium, so we have to collect different sort of chemicals, and the chemicals must be of in a very highly proportionate, right? Even a milligram difference may affect the entire media. So each and every chemical which are used to prepare the media must be weighed at a proper ratio, and according to the given composition it has to be mixed. So we can't use just like that, right? So we have to each and every chemical, whether the macronutrient or micronutrient, everything must be measured before mixing it with the medium. And these are the glass vessels which are used in the plant tissue culture. Generally they use the glass tubes. So this is the simplest glass tube. We can also use large size glass tubes and also the conical flask. And this is the petri dishes. It's a small glass made material which is also widely used in the plant tissue culture. Mostly this is used in culturing the bacterium and fungal species, right? And these are the other equipments which are used in the plant tissue culture. We can able to see the measuring jar, and we can able to see ethyl alcohol. Ethyl alcohol is widely used in the plant tissue culture, and this is most specifically used for sterilizing the materials, right? Some of the materials like steel instruments like forceps or scalpula or scalpels or spatula, all are just wiped with the ethanol dipped cotton cloth, and it is and it is flamed, right? So you can able to see the Bunsen burner or a simple lamp, spirit lamp will be used, and they will be heated in the fire, right, to kill the microorganisms. And cotton is very highly used to cover the glass vessels, right? After the once the inoculation is done, then gloves are used. Then aluminum foil. If cotton is not available, they will use the aluminum foil. And nowadays mostly they are using the aluminum foil as the best choice. And the next is nutrient media. Nutrient media is a very essential part of the plant tissue culture. Without nutrient media, we can't able to perform the plant tissue culture. And there are different sorts of nutrient media available in the market. And the most widely used media all throughout the world in tissue culturing labs are all these four media. And more specifically, Murashi scoop medium, MS medium. We simply call it as MS medium. And that is the most widely used nutrient media in the plant tissue culture. Apart from that, Gambog media, which is also called as B5 medium, then white medium and NITS medium, NITS and NITS medium. So these are the four widely used nutrient media in the plant tissue culture. Media is nothing but the composition of different chemicals, right? In our book, it has been given about the MS medium composition, which is in book page number 111. And these media contains a specific proportion of different types of macro and micronutrients in a correct ratio So each and every nutrient, each and every chemical which is mixed in the media is measured, and according to the correct proportion it is mixed up, right? Even a milligram or microgram level will not vary, right? So only if the proper ratio is given, the result will be fine. And these are the images showing the different types of media. So instead of preparing the medium by ourselves, because if we prepare our medium, only a skilled person can do the medium in a proper level and also in a correct manner, right? So instead of making choosing the choice, we can able to collect the medium from the market, and we can simply add according to the requirement. And moreover, one more substance we can able to see here is the agar. So agar is a very important material. It is a gelling agent, right? So this is a solidifier. If you want to make a medium, so medium is actually of different types. One is the liquid medium, which is in the liquid state, and another one is the solid medium. So if you want to prepare a solid medium, we will add the agar content. So agar is a polysaccharide material which is obtained from the seaweeds, right? So like gelidium or gracilaria, so we can able to get the agar from those algae, and this agar has the ability to tighten the content, to tighten the nutrient material. They will make the medium to get solidified, so that it will be very much convenient for planting the explants, right? Implanting the explants. So if we need a solid medium, they will generally use the agar content. And moreover, there is another type of medium which is called as broth or semi-solid medium, in which the agar content will be added, but it will be in a minimum proportion, so that it won't be get highly solidified, and at the same time it won't be very much liquid. So it will be in between the state. So that is why we call it as a broth or semi-solid medium, right? So all these things what we have discussed now is about the requirements what we need to perform the tissue culturing process in a laboratory. And the next is the steps about the plant tissue culture, right? So there are different steps to perform the plant tissue culture. In our book, it has been given as the 6 major steps. Let us see one by one. First is sterilization. The second step is preparation of media. The third one is culture conditioning. The fourth one is callus formation. The next is embryogenesis and organogenesis. And the final one is hardening. So these are the 6 basic steps which has to be followed in the plant tissue culture, right? So we can able to study one by one in the next video, right? So in today's class, we have discussed about the different types of equipments and the materials which we need in a laboratory for performing the plant tissue culture. And we have discussed about the steps in the plant tissue culture, right? Hope this session will be a useful one. Let us meet in the next episode. Thank you.